for the lengthy title. So I'm Marcin Jąkalski from the Medical University of Gdańsk, and where I work as a postdoctoral researcher. And today I would like to talk a bit about the phenomenon of the loss of chromosome Y and how is it associated with the Alzheimer's disease. So LOI basically is an aneuploidy that removes the entire male sex chromosome, that is around 2% of the human genome. And the LOI occurs during mitosis, presumably as a defect of the regulation of the cell cycle. So this phenomenon has been known for over 50 years, and it is known that LOI is frequent among elderly men, and it accumulates with age. LOI mostly manifests in peripheral blood cells, but has also been found in other tissues. Okay, sure. Okay, thank you. So as a male-specific genetic risk factor, increased risk for pathology and mortality has been associated with the mosaic loss of Y in blood, and this could help to explain why, on average, men live shorter lives than women. So the recent discoveries have shown that cells harboring cells with LOI in the peripheral blood are associated with increased risk for all-cause mortality, diseases such as different forms of cancer and Alzheimer's disease. So we or other scientists have been asking this question, so how come the single mutation can be involved in so many different diseases, pathologies? So our group has recently published this paper where it was shown that LOI has a large impact on transcription and is associated with dysregulation of over 500 autosomal genes. So this paper showed that chromosome Y has in fact a broad molecular role outside of the sex, so challenging the view that this is a genetic wasteland outside of the sex determination. So here we see this effect of LOI in transcription, and we know that epigenetic mechanisms are involved in regulating gene expression. So we wanted to complement this research with such epigenetic data, and we decided on including DNA methylation data. In the previous research, we collected blood samples from patients diagnosed with Alzheimer's disease and age-matched controls from male subjects from Uppsala in Sweden and from Krakow in Poland, for the purpose of estimating LOI levels. And using this data, we selected further a group of 73 individuals for the purpose of correlating gene expression levels with the methylation status. So our analysis focused on two types of leukocytes, namely granulocytes and monocytes, which were sorted using facts, and various types of data were generated for these cell types. So first of all, we confirmed the presence of LOI in both selected subpopulations. We could further see that the high fraction of cells with LOI and a broad range of values in general was observed mostly for the individuals with AD. And depending on the estimated percent of cells with LOI, samples were further divided into two groups, that is LOI and non-LOI group. We use this 30% cutoff, that is where 30% of cells have no chromosome Y. In our data, we could see that AD samples presented a clear bimodal distribution, and this 30% of threshold clearly separated the two peaks of data. So using the Infineon methylation bead chip, we performed genome-wide DNA methylation profiling. So for those of you who don't know what EPIC is, it is basically a chip with 850,000 of probes spread genome-wide, and we first inspected the global distribution of differences in average methylation involving all four studied groups, but the main point of focus here was comparing LOI to non-LOI groups among the AD patients. So while other comparisons show no striking differences, that is, the median change in methylation was around 0, in monocytes, the AD samples not affected by loss of chromosome Y were clearly more methylated than the AD LOI samples. So there was a shift towards the negative values. So we further looked into the inter or intragenic localizations of the CpG probes and their functional annotation. So here the loci that overlapped promoter regions showed a shift toward higher methylation, hypermethylation in LOI. So the more positive values seen on the graph, and this was observed both in granulocytes and monocytes. At the same time, gene-associated methylation changes pointed that among 80 samples, methylation in LOI is lower. So we next continued with statistical analysis of differential methylation to identify LOI-related DNA methylation changes, and specifically for Alzheimer's disease. Among the 80 samples, we could see that hypomethylation in LOI dominated, both in granulocytes and monocytes. Because we performed methylation analysis including other groups than showed here on the graph, we could also identify among the bulk of all DMPs only the ones that were differentially methylated only between AD LOI and AD non-LOI groups. So this shows AD-specific effect of LOI on DNA methylation. For instance, in granulocytes, but not in monocytes, the magnitude of hypomethylation increased significantly among these AD-specific DMPs. So we next look further into genes associated with these identified DMPs. So here we call them DMGs as differentially methylated genes, and in total we found around 2,000 of such genes in granulocytes and around 900 of them in monocytes. And many of these DMGs, that is around 66% in granulocytes and 45% in monocytes, were actually previously associated with Alzheimer's disease. So the sole fact that the gene is differentially methylated does not necessarily mean that there will be some impact on the function of the gene. So hence we look at the RNA-seq data. So our differential expression analysis of AD samples identified over 1,000 of genes demonstrating significant change in expression in granulocytes. And while our differential methylation analysis showed that hypomethylation dominated in LOI, here the LOI-associated effect on gene expression was not that pronounced, as the methylation data would suggest. So we identified examples where the expression methylation data clearly correlated, and this correlation was negative as expected from the canonical model of gene expression regulation by DNA methylation, where hypomethylation in promoter region translates into increased expression. So in monocytes, we also found over 1,000 of DGs, and similar to granulocytes, there was no large shift towards overexpression as the methylation data would suggest. And we also observed DMG-DEG pairs with clear negative correlation between methylation and expression data. So we clearly see effect of LOI both at the DNA methylation level and at the gene expression level. But when we assess to what extent are DMGs and DEGs overlapping, we observed that in granulocytes we only have 62 such genes, while in monocytes it's only 27. That is where we limit DMGs that have change in methylation in the promoter region. So we independently check the functions of these DMGs and DEGs, that is gene ontology terms and KEK pathways in the generation analysis, and we found no overlapping terms. So what could be the link then between these two types of data, two types of analysis? So we further performed gene co-expression analysis using the weighted gene co-expression network, WGCNA, to find gene expression modules. And this helped us to verify similarities on regulation modes between granulocytes and monocytes, but also to link expression data to methylation data. And in both studied cell types, we found modules that were significantly differentially expressed across the investigated samples. Most of these modules were upregulated in LOI. That is, the individual genes that make up the modules were overall higher than in the non-LOI samples. We found 3 modules, here annotated as light cyan and purple in granulocytes and tan in monocytes, that were significantly enriched in gene ontology terms related to immune processes. Hence, our study highlights altered immune cell functions that would link LOI to immune cells directly to the disease mechanism. Although scarce, the DMG-DEG pairs might serve important functions such as hub genes that, for instance, play important roles of transcription factors, inhibitors, or other types of regulators of cells. So next, the high number of DMGs and DEGs, yet their limited overlap, let us hypothesize that further that LOI may affect different modes of gene expression regulation. Many of the significant changes in DNA methylation we identified cannot be linked directly to any specific gene. They comprise between 20 to 30% of DMPs identified in monocytes and granulocytes, and they are located within the intergenic regions, and more than half of the sites lies outside of the annotated CpG islands. So we decided to investigate methylation patterns in these regions. So gene expression can be regulated through action of DNA enhancers or on a higher level through the TADs, so-called topologically associating domains. And when we analyzed histone marks data, we saw that LOI-induced differences in methylation of active promoters region is skewed toward hypermethylation. However, the difference between the AD and control samples, although significant, was not that pronounced as in the case of intergenic enhancers. In contrary, we observed a large shift towards LOI-associated hypermethylation in AD. The average methylation of enhancers peaked around 0 for the controls, and this suggested that LOI has even more pronounced effect on DNA methylation in AD samples than in control samples. So similar pattern was observed when we investigated three-dimensional chromatin maps within TADS comparison of LOI versus non-LOI methylation. Patterns showed that controls display only a slight skew towards negative values, that is higher methylation in non-LOI. In average methylation difference, when analyzing AD samples only, the bias towards the negative values is much more pronounced. The distribution of these stat-bound average differences is significantly distinct between controls and AD samples. So lower DNA methylation means more access to transcription factors or other factors that promote gene expression, or inhibit gene expression. Thus, these results suggest broader changes in gene expression regulation caused by LOI than only limited to canonical promoter-bound model. We additionally include single-cell data to our analysis, and this helps us to investigate influence of LOI on cell subpopulations here limited to monocytes because obtaining 10x data for granulocytes is not feasible. And for each of these cells, we infer their LOI status based on the expression of the Y-linked genes. We compared further the expression in LOI cells from LOI AD patients against the non-LOI cells from non-LOI AD patients. That is the two extremes of the data. And in general, the total number of DEGs based on the single cell data was smaller as compared to the bulk, which could be explained by the lower depth of the single cell data. This analysis also showed that there are some subpopulation differences, as we found different number of DEGs depending on the analyzed monocyte subpopulation. Among the single cell based DEGs upregulated in LOI, we again found enrichment of gene ontology terms related to immune and defense responses. So although this is still an ongoing project, it has already produced some interesting and meaningful conclusions. So far we have concluded that the Y chromosome has a surprisingly broad role. So this is not really a genetic wasteland, as many people treat it. We identified genes that might be involved in pathogenesis of AD and corroborate the main genes already reported as potentially important for this disease. LOI has, in fact, a profound impact on gene transcription, including genes involved in immune system functions. Regulation of gene transcription through DNA methylation might not be limited to genes promoters, as we showed with other types of data. And since we studied immune cells from peripheral blood and not the brain tissue, our results support the role of dysfunctional immune system in the pathogenesis of Alzheimer's disease. So I would like to thank all of my collaborators and thank you for listening to my talk. For a question.
All right, thanks for a really interesting talk. I have two related questions.
Yes.
One of them is basically, I'm curious about where in the pathogenesis of Alzheimer's disease this Y chromosome loss tends to occur. Is it something that's maybe upstream or early of a lot of the other consequences, or is it something that after there's a lot of damage in the body that it occurs downstream? And the related question is, how do you think the blood impact relates to what's happening in terms of the brain pathology. So, most of the Alzheimer's associated pathology occurs in the brain with amyloid plaques and tangles. How could that relate back to Y chromosome loss in the blood?
Thank you for this question. So here we studied blood, of course, the peripheral blood, but there is this brain-blood barrier, so some of these cells can actually, leukocytes can travel through this barrier. But that's actually difficult to answer based on this data, I think. So the one of the hallmarks of Alzheimer's is this inflammation. So with this data, we show actually that with LOI you actually have even more of the increase in this inflammation processes. With the first question, so with LOI, it was shown that the older you get, the more LOI actually, or there's some progressors or non-progressors with LOI, but those who progress with LOI, there is 6 times more chance for them to develop Alzheimer's disease than for the non-LOI and non-LOI progressors. So actually, we don't know whether this is the cause or it happens downstream, as you said, but LOI clearly has this kind of effect. So, but it's really difficult to say whether this could be one of the or the only cause, because we think this has a very pleiotropic effect. So not only that you can narrow it down to a single thing that could trigger some AD related changes.
All right, thanks.